Okay, good morning, everybody. Today's topic is on genetic engineering. I did struggle a little bit to kind of come up with any bioinformatics or clinical informatics angles. And to be honest, the field is expanding so quickly and there's so much information that next week's session is going to be purely devoted to clinical informatics, and there's going to be kind of minimal bioinformatics and clinical informatics. Today we're going to be talking mainly about biotechnology aspects of human genetics, genetic engineering. Next week we'll cover informatics, and then we'll have our ethics debate, which we will not be recording. And then that will be it. That will be it. Before we dig in, are there any questions? Okay, good. So today what we're going to do is we're going to give an overview of genetic engineering. I tweaked the description of the class a little bit. What we're going to be doing is an overview of genetic engineering for primarily two scenarios. One is the development of human gene products, and then the other scenario is somatic cell editing. We'll be looking at a lot of this through the eyes of a clinical case of muscular dystrophy, particularly Duchenne's and Becker's muscular dystrophy. What I want to do is introduce the case, tell you a little bit about muscular dystrophy. I think it's necessary to understand the genetics of the disease before really understanding some of the creative genetic engineering approaches that have been put forward in order to treat it. So we'll introduce the muscular dystrophy case, then we're going to digress and we're going to get into some detail about genetic engineering. And in the context of somatic cell editing, we'll delve into CRISPR-Cas9 technology, and we'll talk about some of the evolving clinical applications of CRISPR. Then when we revisit the clinical case towards the end, what we'll do is we'll walk through some of the clinical trials that are going on with muscular dystrophy, and then that will include some of the CRISPR work going on in muscular dystrophy. My phone started beeping. Can you guys still hear me?
Yes, Bob, I can still hear you.
I can hear you.
All right, great. Thanks. Yeah, my phone seems to be doing something funky. Okay, and so that's what we'll cover today. And just a couple of reminders about homework. Next week, and by the way, thanks for everyone who has been doing some really nice work on this next generation sequencing variant discovery homework. We'll go over it next week as part of the clinical informatics session. And then another reminder, the week after that, we're going to be having an ethics debate, which we won't be recording, but we want to have a class participation session. And really in this class participation session, the way it's going to work is I'm deliberately not preparing any material. I'm not going to be really leading or moderating the debate. And so really the debate is going to rely on what the students bring to the table in terms of, how much we surface various pros and cons. So that'll be in 2 weeks. Okay, so why don't we go ahead and get started with our case of muscular dystrophy. And here we have a case of a 6-year-old boy who was referred for mild developmental delay. His parents noted that he was having difficulty climbing stairs, running, and participating in vigorous physical activities. He had decreased strength and endurance. His parents were fine. He has 2 brothers, they're fine. He has 1 sister, she's fine. No other family members had any similar types of symptoms. When they examined the boy, they found that he had difficulty jumping up onto the examination table. And he also manifested something called a Gower's maneuver, which is what you see here. This is a sequence of maneuvers that somebody with muscle weakness goes through when they're getting up off the floor. They found that he had proximal muscle weakness. He kind of waddled when he walked. His heel cords were tight, and it looked like his calves were enlarged. They measured his serum muscle enzymes, his creatine kinase. His level was 50-fold higher than normal. And so the clinician strongly suspected that he had some type of myopathy, some type of muscle disease, and he was referred to the neurogenetics clinic for evaluation. They did a muscle biopsy on him, and it showed marked variation of muscle fiber size. It showed fiber necrosis. It showed that a lot of muscle cells had been replaced by fat and connective tissue, and it also showed the complete absence of muscle protein called dystrophin. And dystrophin is the protein produced by the DMD gene, and the absence of dystrophin in the setting of all of his other symptoms led to a provisional diagnosis of Duchenne's muscular dystrophy. And he was tested for deletions in the dystrophin gene, and he was found to have a large deletion in that gene. And subsequent testing showed that his mother was a carrier. And so the family was counseled, and they also assessed the mother, because even though this is an X-linked disease, her carrier status placed the mother at high risk for cardiac complications. So this boy has Duchenne's muscular dystrophy, and he has what's called pseudohypertrophy of the calf. And while it looks like he has big muscles there, really what's going on here is the muscle tissue has been replaced by connective tissue and fat. And if you were to do a biopsy of that muscle, what you see here is normal muscle up at the top, and you can see that these are the muscle fibers cut on end. And here, the muscle fibers have been replaced by extensive amounts of connective tissue and fat. And so this is what you see in muscular dystrophy. The gene involved, now there's many different types of muscular dystrophy. The muscular dystrophy that we're going to be focusing on now is due to variations in the DMD gene. And you can have, like with a lot of the other genetic diseases we've looked at, you can have a whole spectrum of phenotypes depending on the nature of the mutation. You can have complete absence of any functional protein, sometimes leading to more severe phenotypes, or you can have partially functional proteins or partial or decreased amounts of proteins where you can have a partial phenotype. And that's exactly what we see in muscular dystrophy due to variations in the DMD gene, where you can have a milder form of disease, and that's what you see here in the middle with Becker's muscular dystrophy, or you can have a more severe form of the disease, and that's what you see here in Duchenne's muscular dystrophy. The picture at the top shows these muscle fibers that are all cut on cross end, where they have been stained for a protein called dystrophin. Dystrophin is the product of the DMD gene, and the protein, as you can see here, localizes to just underneath the sarcolemma, or the muscle fiber's plasma membrane. Okay, so you can see a normal dystrophin, which is all around that cell membrane, just underneath the cell membrane. And in Becker's muscular dystrophy, where you have a partial aberration in the gene, you still have some dystrophin, but you don't have normal amounts. And then in Duchenne's muscular dystrophy, you have an absence of dystrophin. And you can also see here that the muscle fibers, you have much more extensive replacement of the muscle fibers with connective tissue and fat as you progress from normal through the milder Becker's muscular dystrophy to the more severe Duchenne's muscular dystrophy. Okay. The DMD gene is located on the X chromosome. And so you see here a typical X-linked inheritance pattern on this particular pedigree, where females are carriers and males can be afflicted, okay? And so males can pass the disease along to their daughters as carriers, but female carriers can actually have sons that manifest the disease. So your typical X-linked inheritance pattern with a caveat. So here's a female, okay, and this female has two X chromosomes, and at the DMD gene, one of those, one of the alleles on one of the X chromosomes is going to be normal, and one of the alleles is going to be abnormal for the DMD mutation. And you may recall that females have something called X inactivation, where early in embryogenesis, at random, each of the cells inactivates one of the X chromosomes. And as a result, you wind up with a bit of a mosaic pattern in females where some of their cells are expressing genes on one of the X chromosomes, whereas other cells are expressing genes from the alternate X chromosome, and you get a mosaic pattern. And so you see here, when you look at muscle biopsies, here we have a normal male on the left. Here in the middle, we have a boy afflicted with Duchenne's muscular dystrophy, where again we see the complete absence of dystrophin. And here we see on the right, we see a female carrier, where you see that some cells appear to be completely normal with normal amounts of dystrophin. And then you have other cells which are expressing the DMD gene off of the alternative X chromosome, look just like the muscular dystrophy specimen where there's no dystrophin. And so most of the time you see, you don't see any skeletal muscle manifestations of females, but just because of probability, sometimes you will see some manifestation of skeletal muscle weakness in female carriers. The other thing when it comes to muscular dystrophy is that the dystrophin, the product of the DMD gene, is not just being expressed in skeletal muscle, it's also being expressed in other tissues such as the brain, and that accounts for the intellectual impairment that's often seen with muscular dystrophy. And it's also expressed in cardiac tissue, and that accounts for why so many patients with muscular dystrophy develop congestive heart failure and can actually die of cardiomyopathy. And so even the females that don't have any clinical symptoms of skeletal muscle weakness generally can manifest cardiac abnormalities such as dilated cardiomyopathy. And so female carriers of muscular dystrophy, even if they're asymptomatic, generally will have some type of cardiac screening just to assess the extent of involvement of their cardiac tissue. So it's a gnarly gene. It's the largest known human gene. So here we are, we're over on the small arm of chromosome X in the P21 region. And this gene is so big that it actually occupies 1.5% of chromosome X. It's 2.5 million bases. And there are many different what are called isoforms. There are many different isoforms. Some of these, so there's multiple different promoters. You can see this particular messenger RNA starts here at this promoter, whereas this particular messenger RNA starts here at this promoter. There's multiple promoters, there's alternate splice sites, and so you wind up having multiple messenger RNAs, or another way of saying is that you have a lot of messenger RNA isoforms. The primary skeletal muscle transcript is the one that's highlighted here. And this particular messenger RNA has 79 exons. And it's about 14, once all of the introns have been spliced out, the final messenger RNA is about 14,000 bases long. So you start off with this pre-messenger RNA, which is nearly 2.5 million base pairs long. And after all the introns are spliced out, you wind up with a messenger RNA that's now over 100-fold smaller. It's now down to 14,000 bases long. And then that ultimately gets translated into the dystrophin protein. So it's a big, gnarly gene. It's interesting because some of the studies suggest that it can take up to 24 hours just to transcribe one messenger RNA from the DMD gene because it's so big. And some of the stuff that we're talking about now is ultimately going to be very relevant when we start looking at genetic engineering techniques. I think possibly naively when people think about genetic engineering these days, they think of, oh, well, we're going to use CRISPR. But there's no, as you'll see, there's no one size fits all when it comes to applying even CRISPR technology in a genetic disease. Every genetic disease that's been studied and every genetic engineering clinical trial really has some nuance to it, and all of them really require a detailed understanding of the genetics of the particular disease. And in this particular case, what we see in the bulk of Duchenne's muscular dystrophy, about 60% of the cases, we have these large deletions towards the far end of the gene. And what's notable about these deletions, and you can see here that a lot of the deletions that result in the more severe Duchenne's muscular dystrophy look like they overlap the deletions that result in the milder Becker's muscular dystrophy. And really what the key difference between these two is that these deletions that cause Duchenne's result in either the creation of a stop codon or they result in, they either result in a stop codon or they'll result in a missense mutation, or they'll disrupt the reading frame. And so here you have this dystrophin protein where you have at the N terminal, you have the actin binding domain, and at the C terminal, you have the cysteine-rich domain. And these domains of the protein are absolutely critical. But what's interesting is along the middle, where you have the rod domain, you have multiple repeating units. And so in the milder forms of Becker's muscular dystrophy, if you have a deletion, but you don't have a frame shift, or if you have a deletion and you don't introduce a premature stop codon, you still have a dystrophin protein, although a chunk of the rod domain may have been truncated. But if you don't have the premature stop codon, or if you don't have the disrupted reading frame, then you still wind up having a functional cysteine-rich domain, and therefore the protein remains largely functional. So while both Becker's muscular dystrophy and Duchenne's muscular dystrophy are characterized in many cases by these large deletions occurring near the tail end, really what distinguishes them is that the more severe Duchenne's muscular dystrophy, the deletions introduce a premature stop codon or they disrupt the reading frame, and as a result, in addition to truncating this repeating units of the rod domain, they also wind up screwing with the cysteine-rich domain, and that then results in a much more severe phenotype. And that's going to be very important when we come back to genetic engineering, because I'll give you a heads up, this particular gene is so big that it results in major challenges for genetic engineering. This gene is way too big to simply reintroduce a brand new gene into the cell. Okay? If we look again at the dystrophin protein, what we'll see here is the N-terminal of the protein and the C-terminal of the protein. The N-terminal of the protein is critically important for interacting with the actin and the myosin framework, the contractile mechanism of the cell, and somehow anchoring that to the cell membrane or the sarcolemma, to the dystroglycan-sarcoglycan complex of proteins that then connects to the outside world. So this dystrophin protein, whose function is not completely known, does appear to be somehow bridging between the proteins embedded in the cell membrane and the actin contractile framework of the cell. And when you have aberrancies in the dystrophin protein, it leads to disruption of the cell membrane. The cells die. The cells ultimately then get replaced with connective tissue and fat. But the other thing to note about this particular picture is while you have these key functional domains on either end of the protein, you have these large repeating units. And it turns out to be not as functionally devastating to simply remove a repeating unit as it would be to disrupt the N-terminal or the C-terminal. And so we're going to come back to that when we talk about genetic engineering. We can simply remove one of these repeating loops, and that can improve function, even though it doesn't necessarily fully restore function. Okay? So that's our introduction to Becker's and Duchenne's muscular dystrophy. We're going to digress now. We're going to start talking about genetic engineering. And then after talking about genetic engineering, we will come back to muscular dystrophy and look specifically at how some of the genetic engineering technology is being applied to this particular disease. Before delving too into genetic engineering, just a couple of introductory slides. People may wonder what genetic engineering is, and while the definitions may not fully align, I thought for the scope of this class I would just lay it out clearly that, for our intents and purposes, what we're going to be talking about with respect to genetic engineering is the direct manipulation of an organism's genome using biotechnology. In other words, we're mucking with your genes. We're literally mucking with your genes. It doesn't necessarily mean that we're introducing new genes. We could be doing something to cut or disrupt your genes, but somehow we're directly manipulating an organism's genome using biotechnology. And this can be used to change the genetic makeup of cells. It can include the alteration or the transfer of genes within and across species boundaries. So we're not talking about engineering required to, make a DNA chip. We're not talking about engineering required to introduce an interfering RNA molecule to treat a disease. We're specifically here going to be talking about the biotechnology that's used to directly muck with an organism's genome to alter it. Okay. And there have been gene therapy trials now going. There have been gene therapy trials now going on for years, and this is a kind of just a set of examples of some of the inherited diseases that have been treated by gene therapy of somatic tissues, and we're only going to be talking about genetic engineering of somatic tissues here. We're not going to be talking about gene therapy, excuse me, of germ cells, just of somatic tissues. One of the things you'll see as you look over this is a wide range of viruses that are used. The viruses are transducing the cells. What that means is the viruses are carrying with them genetic information that leads to a change in the host cell's DNA. They're introducing foreign DNA into the host cell. In other words, they are transducing the host cell. So you'll see that there's a number of different viral vectors that are used here: retroviruses, lentiviruses, adeno-associated viruses. You'll also see that there are a number of different transduced cells, stem cells, hematopoietic stem cells. In this case here, the adeno-associated virus vector was actually directly injected intramuscularly. So we'll also see examples of in vivo applications of gene therapy. And one of the things, so we're going to cover a number of these aspects. We're going to be talking a little bit about the different types of vectors and what are some of the pros and cons of the various types of vectors. And we're also going to be talking about the different types of cells that can be transduced, whether they can be transduced in vivo, in other words, by a direct injection of the virus into the body, or outside the body, ex vivo, we'll be talking about as well. And so these are some examples of diseases that have been treated by gene therapy. This is about the most up-to-date information I could find. There's well over 2,000 clinical trials. Clinicaltrials.gov, if you search for gene therapy, you'll find that it lists over 5,000 clinical trials. The bulk of gene therapy clinical trials are being directed at cancer diseases. We'll talk about a couple of innovations in genetic therapy for cancer because this is really where such a large focus of activity is taking place. Then for monogenic diseases, and this would include Duchenne's muscular dystrophy, and we'll come back to that, and I'll give you an example of how gene therapy is being used to try to combat HIV disease. And you can see there's a number of other diseases that are being studied with gene therapy. And vectors that are often used are viruses. This particular reference, they call out CRISPR-Cas9 as a specific vector, which is a little bit misleading because oftentimes the adenovirus vector, the gene that the adenovirus vector is actually carrying in is a CRISPR gene. But in this particular graphic, they call out CRISPR being used as the vector in 7 of the cases. N would equal 7, or 0.3%. And in clinicaltrials.gov, they show out of about 5,000 trials for gene therapy, they show CRISPR being used. They have about 10 CRISPR trials. So there's not a whole heck of a lot of active human trials using CRISPR, but it appears based on these websites that there's somewhere around 10.
Hey, Bob, wondering if they publish the success rate of these trials.
There's a couple of things. One is you have summaries about these trials, such as what you have here. Mm-hmm. And so there are summaries of the trials. Then, of course, for each individual trial, there'll be publications about it. I don't have a slide here other than this one, which kind of summarizes a number of trials. But those types of summary tables do exist out there on the internet.
Great.
Bob, on the previous slide, what is gene marking, and what kind of genetic manipulation studies are done on healthy volunteers?
Let's see. You know what? I could guess, but I can't tell you for sure. Maybe that's Randy, right?
Correct.
Yeah, Randy. Maybe it's better if I just get back to you on that, because I think I know, but I'm not positive.
Okay, thank you.
That's slide 16. Okay, so we're going to be talking about kind of two big buckets of genetic engineering. One big bucket is going to be the application of genetic engineering to create human gene products. And then the second big bucket is going to be somatic cell editing. If we look at this first big bucket, human gene products, this begins to introduce you to the notion of cutting and pasting genes. And I think, people are very familiar with the term CRISPR, but I think it's important to know that CRISPR is a nuclease. CRISPR is an enzyme that can cut DNA, but there's hundreds of enzymes that can cut DNA. And enzymes that can cut DNA, one of the things they have generally in common is they each have a specific recognition site that when they see that recognition site, they can glom on and cut. And so the EcoR1 nuclease, for instance, finds sequences where you have a CTT-AAG. And wherever it finds a sequence that's a CTT-AAG, it cuts. And as you can see, it makes kind of a staggered cut. And it leaves edges with overhangs, or what are called sticky edges. And that allows you to take, for instance, human DNA and cut out a gene of interest, for instance, the gene for insulin. It allows you to take DNA out of a bacteria. In this case, this might be a plasmid from E. coli, and you cut the plasmid using the same nuclease, the EcoR1 nuclease, and what you wind up with is now having a plasmid with sticky ends, the human gene segment with complementary sticky ends. You can hybridize the two together, and you can then seal the deal with a DNA ligase. One of the things also that this points out is what is called a palindrome. A palindrome in DNA is where you have the sequence that reads the same forward as it does backwards. So here you have CTT-AAG, and on the reverse strand you see CTT-AAG. Sometimes we think of palindromes, like my name Bob, B-O-B. You can read it forwards and backwards the same way, but the definition of palindrome in DNA is a little bit different. It's where the forward strand and the reverse strand are the same. And so these nucleases are searching for specific palindromes, and you have nucleases for a lot of different patterns. You cut one, you cut the other, you hybridize them together, and now you wind up with, in this case, a recombinant DNA molecule. It's the bacterial plasmid to which a human gene of interest has been introduced. So again, this particular EcoR1 nuclease, I think maybe people confuse that with, they think that every time we cut DNA, we're using CRISPR. And just to make the point again, there's hundreds of different nucleases that cut DNA, and each one has a specific target sequence where it will cut that DNA. So if we wanted to develop some human gene products, what we can do is we can take a bacteria and we can remove its plasmid. And a plasmid is kind of like a chromosome, but it's a little circle of DNA. You can kind of think of it also as being analogous to our mitochondrial DNA. You can remove that plasmid and you can take the human DNA and isolate the gene of interest. You can cut that gene out. You can cut open the plasmid. You can insert this gene of interest into the plasmid. You can re-anneal these with your DNA ligase, and then you can get the plasmid back into the cell. Once you have that plasmid with the human gene back in the cell, you can now allow this bacteria to grow, And as this bacteria grows, not only is it transcribing its native genes, but it's now also transcribing and translating the human gene that was put into it. And this technique has been used to create a lot of human gene products. Shown here, for instance, would be growth hormone or thrombolytic agents. And this next table here gives you an example, just some of the human gene products. And Rafiq, there would also be more comprehensive tables if you wanted to find, for instance, a list of all FDA-approved recombinant DNA gene products. I thought this table was kind of nice because it also shows the chronology. Insulin was the first recombinant DNA that was licensed for use, and you can kind of see sequentially a number of them. The factor 8, was quite significant because up until the time that we were able to produce factor 8 using recombinant DNA, we were having to treat hemophiliacs with donor-derived plasma, and that accounted for, I think it was upwards of 50% of hemophiliacs at some point were HIV infected, and their lifespan, the average life expectancy for a hemophiliac was dramatically diminished. Now, of course, we have recombinant factor 8, and that's largely obviated the problem.
Thanks, Bob.
Mm-hmm. Okay, so that's really all I want to say about human gene products. I really now want to start to delve into somatic cell editing and work our way into a discussion of CRISPR. This picture here is super high level, just to kind of set the stage. It's very simplified. There's tons of techniques that really are not adequately reflected by this particular picture, but it's mainly intended to serve as a gentle introduction to the notion of somatic cell editing. And what you see here is we start with a retrovirus. We're actually removing some of the genes from the retrovirus, and these are genes that would be, for instance, necessary in order for the retrovirus to replicate. It's one of the safety features. The retrovirus can't replicate, and in its place also there's a limited amount of DNA that fits, or RNA that fits into a retrovirus, and by removing some of the existing genes, we actually make a little bit more space to insert the genes of interest, in this case a therapeutic human gene. The other thing that's pointed out here is that not only are we inserting a gene, but we're inserting a regulatory element. You may recall back our discussion about thalassemia, where some of the manifestations of the disease were due to an imbalance between the number of alpha chains and the number of beta chains in the hemoglobin molecule. And if you had too many of alpha chains, or if you had too many beta chains, they would precipitate, and that would lead to cell damage. So oftentimes in genetic engineering, it's not simply a matter of introducing the correct gene, but it's also important to include the regulatory elements that help determine the amount of gene that will be produced, that will be transcribed, also the tissue in which that gene will be produced. So we put the therapeutic gene and regulatory elements into the retrovirus. We then replace the retroviral genes with the therapeutic human genes, and we put them back into the virus, and then we allow the virus to infect the cell. Now, in this particular case, we're dealing with a retrovirus, which is an RNA virus. The RNA virus has a reverse transcriptase enzyme, and that changes the RNA back to a DNA, and that DNA then actually integrates itself into the human cell's DNA. And now that that gene has been reincorporated back into the human cell DNA, it, theoretically acts as if you had that gene all along. That gene then can produce the therapeutic gene product, and of course, every time this cell divides, all of the daughter cells have the gene, and there you are, you're cured. Now, with these retroviruses, one of the challenges, in addition to the fact that you don't have a very large payload, is you don't have control over where this DNA inserts. And so in some of the earlier experiments with this type of process, the DNA inserted itself in a place such that it activated an oncogene. And so there have been a handful of cases of patients that have developed cancer, some of which have died from cancer because the virus, the viral DNA, and the reverse transcribed DNA inserted itself such that it stimulated an oncogene. So there's a number of issues that are being addressed, and the amount, where and when the particular gene gets transcribed. You have a small payload. You have the potential for insertional mutagenesis. These are issues that all have to be addressed. If we look at a little bit more detailed picture of somatic cell editing, that's what we see here. And again, this is a more comprehensive picture, but there's also, I guess I would also say this picture is not all-encompassing. We can, for instance, introduce, and we haven't talked about CRISPR yet, and this is going to be a little bit confusing, but you can introduce CRISPR protein into a cell. The CRISPR protein can alter the genes, and because it's altered the genes, it meets our definition of genetic engineering. So you're not always having to introduce a gene into a cell. And this picture here shows that genetic engineering kind of is introducing a gene into a cell somewhere or the other. But there are techniques that don't require the introduction of a gene into a cell in order to alter the genome of that cell. So with that caveat in mind, let's take a look at this picture. What we see here is the sequence that we want to introduce. The blue is the gene, but now it's not really the gene as the gene exists in a human cell. This is really what this is the gene with the introns removed. So imagine, if you will, in order to create this gene that we're going to be inserting into a cell, you take the messenger RNA, which is now just all the exons glued together with the introns spliced out. You take that messenger RNA, you apply a reverse transcriptase to it, and you wind up getting the coding DNA. And now that's good enough, because if that can be transcribed and translated, you wind up getting the equivalent functional product. Plus, it's a heck of a lot smaller than the complete gene, and we have some pretty tight size limitations in today's genetic engineering. So what this gene really is the coding DNA, and then it's also got the promoters and enhancers that govern, or that at least attempt to govern, the amount of expression and the tissue specificity and the timing of expression, so that you get ideally the right amount in the right place. This gene can be then introduced into a virus, and then that virus can be directly administered to the patient, just like a shot, given in vivo. Alternatively, we can harvest cells from the patient. For instance, we might harvest hematologic stem cells, and we might then infect those cells with this virus and transduce the stem cells ex vivo in the lab, and then take the transduced cells and reinfuse them back into the patient. And then, because these are stem cells, they would they would find their spot, they would continue to replicate, and you could effect a cure that way as well. In addition to viral delivery mechanisms, there are a number of non-viral delivery mechanisms. When we look at the clinical trials, they're all based on viral delivery mechanisms, but there are a number of non-viral delivery mechanisms which are evolving as well. This one here shows that the desired gene sequence can be put into a plasmid, and there are techniques that can induce these cells to take in the plasmids through a process of endocytosis. And then the plasmids, having been taken up into the cytoplasm of the cell, a certain percentage of them will make their way into the nucleus of the cell. And researchers are constantly looking for more efficient ways to deliver the desired gene into the nucleus of the cell where it can have its effect. So we can either transduce cells ex vivo, or we can transduce cells in vivo, and we can do that using viral vectors. We can do that doing non-viral vectors. And so I want to digress a little bit and talk a little bit these vectors, just to kind of give you a little bit of an understanding about the different types of vectors that are out there. And then I want to come back to talking a little bit about what type of cells are we generally trying to transduce. What cells are we transducing in the lab? What cells are usually the targets of transduction in vivo tests? And then from there, I think once we kind of understand that framework, then what I want to do is talk about, okay, what the heck is this CRISPR stuff, and where in this whole process is CRISPR being used? Or it might be more correct to say, where in this process today is CRISPR not being used or not being envisioned to be used? Okay. So a little bit about non-viral vectors. I think that the science is evolving pretty quickly, and I haven't found any active clinical trials that are using anything other than viral vectors on clinicaltrials.gov and on that other site. They're all based on viral vectors, but in the lab, when people are experimenting with transducing cells, people are using plasmids, they're using gene guns, electroporation, where you zap the cell with electricity in order to have it take up foreign DNA. Liposomes and nanoparticles are kind of interesting. These are particles that are fused. They fuse with the cell membrane, and then they release their contents into the cytoplasm. So a number of different technologies are evolving. Really, the objective here is, how do you get the DNA into the nucleus of the cell, or how do you get the CRISPR into the nucleus of the cell where it can do its thing? Far more commonly, people are using viral vectors, at least in all of the human clinical trials. People are using viral vectors, and there's really 4 main viral vectors that I thought would be worth talking a little bit about, just looking at some of the key features. And some of these key features are the packing limitations. And so the adeno-associated viruses are very popular because they integrate into the cell without requiring cell division. But on the flip side, they have a very tight packing limitation. You can only insert 5 kilobytes, 5,000 bases. The retroviruses, on the other hand, you can put in a big package, but the retroviruses have to integrate into the host DNA and can only integrate into the host DNA when there's cell division. The adenoviruses and the adeno-associated viruses don't actually integrate into the host DNA. Rather, what happens is the viral particles live in the nucleus in a form called episome, as episomes. So viral particles live in the nucleus. They don't actually have to be integrated into the host DNA. And these are attractive in vivo studies, for instance, when you can get the virus to be taken up by cells that have a very prolonged G1 phase, like a muscle cell or a hepatic cell or a neuron. Because if you can get, for instance, the muscle cell to take up the genetic material of the adeno-associated virus, and if you can get the DNA from that virus to work its way into the nucleus and live there as an episome, that cell will live for years. And the episome will be transcribed and its gene products will be translated just as if it were a gene that were on a chromosome. So the episomes from the adenovirus and the adeno-associated virus can be very effective in transducing long-lived cells for in vivo genetic engineering. Retroviruses actually then can permanently transduce cells. And so, for instance, if you are trying to muck with a hematologic stem cell ex vivo in the lab, you would want to use something like a retrovirus because you would want its gene product to be fully integrated into that stem cell's DNA so that when you re-inject that stem cell back into the body, every time that stem cell duplicates, all of its sister progeny are going to carry that particular genetic alteration. And so these are some of the key factors is, can you use these non-integrating viruses, particularly for long-lived cells, or do you need to use viruses that actually integrate? And of course, coupled with that are the size limitations, which are really pretty severe. These are like 8,000 bases. And remember, the size of the DMD gene is 2.5 million bases, right? So there's no way you're going to be using a viral vector to move an entire DMD gene into a cell, given our current technology. Immune responses are also important in that there was an early death in a clinical trial using an adenoviral vector. The patient wound up dying because of the massive immune response that was mounted against the adenovirus, whereas we don't see so much immune response with other viral vectors. Okay, any questions about the viral vectors? Okay. A little bit
Bob,
about stem cell. Yeah.
Was flu virus ever used? I thought I had once heard something about flu viruses being used.
Yeah, I did see flu virus used. I thought I saw flu virus used. I recall it's used. I don't know if it's being used now. We can look at this. If you go to this website, it has a much more detailed table about all of the known vectors being used today. So we can check that. Off the top of my head, I'm not sure. Okay, so let's talk a little bit about stem cells. Probably people are familiar with the notion of a stem cell. Stem cells have these unique capabilities, unique in that no other cells in the body can do this. Let's say no other cells in the adult body can do this. Stem cells can self-renew. So you can have a stem cell and it can split into two identical stem cells. Alternatively, a stem cell can split into two daughter cells that are more specialized, more differentiated. Stem cells have this third capability where they can have an asymmetrical division where one of the daughter cells is an identical stem cell and one of the daughter cells is a differentiated stem cell. So stem cells can really divide in three ways. They can have a symmetric self-renewal division. They can have a symmetric differentiation division, or they can have an asymmetric division where you get another stem cell and you get a differentiated cell. Differentiated cells that are downstream from stem cells no longer have the capacity for self-renewal. Only stem cells have the capacity for self-renewal. Once you've gone past the stem cell compartment, cells further differentiate with each division until they reach their final differentiated state. And we see this again in a little bit more detail on this slide when we look at hematopoietic stem cells or hematologic stem cells. Stem cells can self-renew, but only stem cells can self-renew. When you get downstream, the only thing that the progenitor cells or the lineage-committed precursors or to some extent mature cells can do, the only thing that they can do is divide, and when they divide, they further differentiate. And somebody drew an analogy between this cascade and a boulder rolling down a hill. It's not to say that it's absolutely impossible that a granulocyte progenitor could in fact become and go back upstream to become a common lymphoid progenitor. But as far as we know, that doesn't happen. As these cells progress and differentiate, specific epigenetic marks are being laid down. There are specific transcription factors being applied, and it looks as though this is really, for the most part, a downstream process where cells once a cell has become, for instance, a lineage-committed precursor, it can't work its way back up to become a progenitor cell. Theoretically, it could because the DNA is all there, but in practice, it would be like pushing a boulder back up a hill. The energy requirements to do that would be prohibitive to the point that we don't see it. So as we progress from stem cell to progenitor cell to precursor cell to mature cell, we see greater and greater differentiation, and we see complete loss of self-renewal ability once we leave the stem cell compartment. What that means is, for instance, a progenitor cell will not divide to create two new progenitor cells, will not divide to create two new common lymphoid progenitor cells. Rather, when this cell divides, it's going to create two downstream progenitor cells. Excuse me. The overall proliferative activity of cells, stem cells can be very sluggish. They don't divide all that often, especially in some of the deeper stem cell compartments. Progenitor cells, on the other hand, are highly active. They proliferate quite actively. And as you work your way down to precursors to mature cells, you reach a point where you see no proliferative activity, and these mature cells simply don't divide. They're a terminal state. There's an exception here, though, and that is the memory B cells and the memory T cells. Memory B cells and memory T cells retain the ability to self-renew and to proliferate. Okay, so particularly in response to, a couple of years goes by, you're re-exposed to an antigen, your memory B cells and your memory T cells can replicate, they can self-renew, they can proliferate. So when we look at stem cells, oftentimes stem cells are a very nice target when we think about genetic engineering and transduction, because if you can modify a stem cell, you're in effect modifying all of these downstream cells. On the other hand, sometimes the efforts genetic engineering efforts are directed at T cells, because T cells can in fact self-renew. They can continue to proliferate. Okay. Now, I said that cells don't go back upstream, that the energy requirements to go back upstream would be high, but experimentally, it turns out that we can induce the development of stem cells. And so, for instance, we now know that we can take adult fibroblast cells or other cells, and we can supply specific transcription factors. And there's various transcription factors in various programs that can be used to modify these fibroblast cells into what are called induced pluripotent stem cells. And these cells are pluripotent. And let me just back up a minute. The hematopoietic stem cells are called multipotent, in that a hematologic stem cell can produce any of these cells, but a hematologic stem cell isn't going to create a nerve cell, and it's not going to create a liver cell. So a hematologic stem cell is already partially committed, and therefore it's multipotent, whereas a pluripotent cell is a step up from the multipotent cells, and an induced pluripotent cell can be induced in the lab to form cardiomyocytes or fat cells or neurons or hematologic stem cells. Just to finish that discussion, above pluripotent would be what's called totipotent, and the only totipotent cell that we know of is a zygote. And totipotent means not only can it produce any target tissue, but it can actually give rise to a whole living organism. So we don't have any technology for totipotency. The only totipotent cell we know of is the zygote. Downstream from there is the pluripotent stem cell, which we can induce in the lab. And as you can imagine, harvesting a fibroblast cell, if in fact this really works out and the bugs can be worked out, imagine you do a skin scraping, and now all of a sudden you can do genetic engineering on these induced pluripotent stem cells. I can then have these cells, I can apply experimental conditions that allow these particular cells to mature into cardiomyocytes, and then these can be injected back into the heart, or these can be injected back into the muscle. And we will be talking about CRISPR activities for Duchenne's muscular dystrophy, and I'll give you a heads up that some very futuristic thinkers are already planning experiments where they apply genetic engineering to induce pluripotent stem cells as a way of treating not only the skeletal muscle problems with Duchenne's muscular dystrophy, but also the cardiomyopathy that is often the cause of death. So we'll come back to this notion of induced stem cells because it offers some pretty futuristic and pretty amazing and phenomenal therapeutic options in medicine. Okay, so let's talk about CRISPR. I'm guessing that everyone on the call has heard of CRISPR, that you kind of have a sort of idea of how it works. And what I want to do is take you into it in some detail so that you really have an understanding of what it can do and you have an understanding of what it can't do. And from there, when we talk about how CRISPR is being applied in clinical trials, I think you're going to walk away, I hope, with a far more intuitive gut sense of where CRISPR is going to come into play. So CRISPR is part of the bacterial immune system. It was discovered in bacteria, and a couple of very smart people quickly realized its potential applicability for genetic engineering and for human trials. Really, at a high level, the most important thing to know about CRISPR-Cas9 is that it enables precision cuts in DNA. And that's the main thing I want you to take away from this discussion of CRISPR, is it allows you to, you have this genome with 3 billion base pairs, and you may want to do one little cut way over here, and CRISPR, at least with some caveats that we'll talk about, enables you to target your cut to exactly where you want in the genome, okay? So if we look at this picture, the pink ribbon here would be just our normal human DNA sitting in our cell. And you can think of CRISPR really as being two parts. It's this Cas9 enzyme, and this is the cutter, okay? And then the guide RNA, which hones in the Cas9 cutter to just the right spot. So the CRISPR-Cas9 system really is a complex. You have the Cas9 cutting enzyme, which in nature forms a union with the guide RNA, okay? So these two things form a union, and then it floats around, and if the guide RNA that's part of this CRISPR-Cas9 happens to find a matching gene sequence, then the CRISPR-Cas9 locks in and cuts, okay? And just to give you a heads up, we can specify exactly what we want the guide RNA to be. So this guide RNA is about 20 bases long, and imagine now we can specify whatever we want this guide RNA to be, and so anywhere we want to target, we find out, okay, here's the 20 bases, and that's the place we want to target. Okay, let's go engineer our guide RNA to target that spot. And already you can begin to see some caveats, right? We have a lot of repetitive regions in our DNA. And so if you designed CRISPR, if you only wanted to target one specific area, but it happened to be a repetitive area, well, the thing is, if you use CRISPR, everywhere you find a match, you're going to get a cut. Now, on the other hand, repetitive regions typically are not occurring in exons. And most of the time when we want to do genetic engineering, we're targeting exons. And we don't see hardly any repetitive DNA in exons. And when you have a guide where the specificity of the match is determined by a sequence of 20 bases, well, you pretty much can target almost any exon sequence you want in our genome with very high fidelity. And so CRISPR-Cas9, with some caveats that we're going to talk about, enables you to make precise cuts pretty much anywhere you want in our genome, and you do that by modifying this particular 20 base pair sequence of the guide RNA. What CRISPR actually stands for and what Cas9 actually stands for, I'm going to tell you, but I don't want you to dwell on that because this is just, someone tried to come up with a clever acronym to make it stick in your head. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and Cas9 just happens to be, there's a gene for the Cas9 enzyme, and the particular gene for the Cas9 enzyme happens to be adjacent to the CRISPR region of the bacterial genome. So it was called the CRISPR-associated protein 9. Okay, so that's about all I want to say about what CRISPR and Cas9 stand for, other than I want to show you, in the slide after this, I want to actually show you a little bit more about what these palindromic regions are to help you explain how we discovered CRISPR. But before going there, I think this slide is a little bit difficult to understand, yet very, very important in understanding what you can do and what you can't do with CRISPR. So think of CRISPR as doing a very precise cut, okay? Again, think of CRISPR. It enables precision cuts in DNA. That's what CRISPR does, okay? With some caveats, but think of CRISPR, it does precision cuts. So we can make this double-stranded break with some caveats wherever we need to in the genome. The way that a double-stranded break heals is not part of CRISPR, it's part of the cell's normal double-strand break repair mechanism, okay? And it's important to understand how cells repair double-strand breaks so that you understand CRISPR is making the breaks and then the cells are healing those breaks, okay? And the cell heals those breaks by one of two processes. One of them is called non-homologous end joining, and one of them is called homology-directed repair. And I know that this is getting a little bit deep and detailed, but by going through this, you're going to understand exactly what results from a CRISPR-based cut. If you make a cut with CRISPR and you simply allow the ends to join back up, to reconnect, and that process would be called non-homologous end joining. You make a cut and you just let these ends join back up again. In the process of these ends joining up again, there might be a little bit of a disruption. There might be a couple of bases that are lost, or there might be a couple of bases that are added. So when CRISPR makes a cut and these two ends are allowed to simply reconnect, you will generally get some type of small mutation at the place where the repair, at the place of repair. You might get a small base substitution, you might get a small deletion, you might get a small insertion. So if your goal is to introduce a small mutation, then you can apply CRISPR, do the cut, and just let the cut heal on its own. And in a percentage of cases, the healing of that cut will result in variable length insertions or deletions through a process, just a basic process of non-homologous end joining. Alternatively, if you do that cut, but there happens to be a donor template present, then the repair can be directed based on that donor template through a process called homology-directed repair. So imagine, if you will, here's a cut, and this is a homologous piece of DNA. So this strand of DNA connects right here because it's homologous. This piece of DNA connects right here because it's homologous. And then the gap between these two will be filled in based on the donor template. Okay, so, and this is common when we have recombination during meiosis. Recombination requires there to be double-strand breaks, and those double-strand breaks are healed using a process of homology-directed repair. The advantage of homology-directed repair is that it results in a very precise insertion or modification, and that's why cells use homology-directed repair during recombination and meiosis. The tricky part from a genetic engineering perspective is that it's very difficult to supply donor templates. if I squirt some CRISPR into a cell and I squirt some donor template into the cell, it's not a given that in all cases where the nuclease cuts, the donor is going to be available. And so even if I squirt CRISPR and donor templates into a nucleus, there will be a number of places, where the donor simply didn't happen to be contiguous at the time of the repair, and the non-homologous end joining took place. So donor templates, the advantage is you wind up getting very precise insertions or modifications. The challenge is it's technically very difficult to supply a donor template and to achieve a high degree of transduction with homology-directed repair. If it were feasible, it would be golden because you could do very precise editing. And so I think it's important here to note that what CRISPR is doing is very precise breaks, but for most applications, you're getting imprecise repair. And so the exact mutation that you're inducing when the CRISPR break repairs generally is imprecise. Okay? Ideally, it would be precise, but the current technology means that it's imprecise today, even though you are able to make a fairly precise cut. Make sense? Okay.
Yes.
I'll take that as a, okay, thanks. Okay, so let's take a step back and really delve into what CRISPR is. By understanding really what CRISPR is, you'll have a much better understanding of how we can manipulate CRISPR to do exactly what we want. On the left is native CRISPR in a bacteria, part of this bacteria's adaptive immune system. And here we see the bacteria's DNA. Okay? And what was discovered was that there are these clusters of palindromes. Okay? So I guess you could say these were clustered, regularly interspaced, short palindromic repeats. Oh, CRISPR, okay, right? So we found that there were areas on the bacterial chromosome, bacterial DNA, where we found these clusters of palindromes. And in between, the regions in between are referred to as protospacers. And as it turns out, what these protospacers are snippets of foreign viral DNA that had tried to invade the bacteria. When the virus tried to invade the bacteria, if it won the war, was able to cut up the viral DNA or RNA and insert fragments into these spaces between the CRISPR elements as a way of remembering viruses that have been encountered before. On another spot on the bacterial genome, they also have a gene that produces another RNA called tracer RNA. And at another spot on the bacterial genome, they also have a gene that produces the Cas9 enzyme. Okay? So what happens is the protospacer is transcribed, and so we have now RNA or CRISPR RNA that's specific to an invading virus. We have tracer RNA. The tracer RNA is necessary for binding the complex of tracer RNA and CRISPR RNA to the Cas9 enzyme. So the tracer RNA glues together with the Cas9 enzyme, and it also has with it one of the protospacer RNAs. And now, should that virus try to invade again, the CRISPR-Cas9 system is going to recognize the viral DNA and cut it up. And it was recognized that, you know what, we could modify the CRISPR RNA. This is a 20 base pair sequence, but you know what? We could make it an arbitrary 20 base pair sequence. And so now on the right, we're building our own CRISPR-Cas9. We genetically engineer our own 20 base pair sequence of CRISPR RNA. The tracer RNA is fixed. That's really just there to glom on to the Cas9. The Cas9 gene is fixed. We just want that Cas9 protein. We create a guide RNA where we have the unique sequence and we have the constant sequence. That then gets complexed with the CRISPR-Cas9, and we can now use CRISPR-Cas9 to hone in on any specific part of the genome and do its cutting. Okay? I might point out here that the system, another caveat to the system is the CRISPR RNA is 20 base pairs long. And for the most part, it's going to find and hybridize a complementary sequence of DNA that's 100% complementary. But we know that there are off-target complications with CRISPR where, for instance, there might only be a match of 19 of the bases. And CRISPR-Cas9 will connect long enough, even though it's not a perfect match, it will connect long enough for the Cas9 enzyme to cleave the DNA. So we do know that we do not see, for the most part, the 20 base pairs have to match the target. But we do see a number of cases where even with a partial match, we will see cuts. And that's been of major concern, particularly when we start talking about using CRISPR-Cas9 cutting for use in human trials because of uncertainty around off-target effects. And let's see. I think this is the last kind of real technical slide about CRISPR. And I kind of sort of feel like, you know, what the heck, it's good to understand CRISPR at a little bit more technical level so that you know the strengths and the weaknesses. We see here again the Cas9 enzyme, and we see that it's glommed on to its guide RNA, and that guide RNA is the constant tracer RNA with the CRISPR RNA, where we can modify the 20 bases based on wherever it is we want CRISPR to cut. With a caveat, the host, the target DNA, we need to find a spot where these 20 bases occur immediately next to what's called a PAM site. PAM stands for a protospacer adjacent motif. So in this particular case, CRISPR is only going to cut where it finds 20 complementary bases immediately adjacent to an NGG motif. And N stands for any nucleotide, GG. Okay, so if the NGG weren't here, then even though these 20 bases paired exactly with these 20 bases, there wouldn't be a cut. You only get a cut where the 20 bases match and that match is adjacent to the protospacer adjacent motif, or PAM site. Now, keep in mind this Cas9 enzyme is a bacterial enzyme, and most commonly we're using Cas9 extracted from Strep pyogenes. But there are Cas9 proteins that can be extracted from other bacteria, and depending on the bacteria you obtain the Cas9, the PAM site varies. So for Strep pyogenes, the PAM site is an NGG. The reason that this is significant is because you may want to do a cut over here, but you can't do that cut unless you're cutting adjacent to a PAM site that's recognized by your particular Cas9. When CRISPR finds a complementary 20 bases adjacent to the PAM site for that particular Cas9 enzyme, the cutting is taking place just a few bases contiguous to the PAM site. Okay? And so that's where the double-strand cutting is taking place, right next to the PAM site. Okay? And that's about as detailed as I want to go in explaining what CRISPR is, but there has been a lot of effort to say, well, okay, we understand that CRISPR does some wonderful things, but now we understand that CRISPR-Cas9, and by the way, there's other Cas proteins, commonly Cas9 proteins are used, but there's a whole number of Cas9 proteins that have different properties. But now that we understand some of the strengths and limitations, we can say, well, can we make further modifications? And so there is a lot of work going on right now actually modifying the Cas9 nuclease in order to address some of the limitations of the CRISPR system. So here's native CRISPR right here, where you find the CRISPR RNA matching up with the homologous 20 bases, and that's adjacent to the PAM sequence, and here's the cutting. Well, it turns out that Cas9 really has two nuclease domains. It has what's called an RUVC cutter and an HNH cutter. And we can modify the Cas9 protein to disable one or both of these cutters. So if we disable the RUVC cutter, we wind up with what's called a nickase, where we're just making a single-stranded cut. Alternatively, we can disable the HNH, and we can have a nickase that cuts the other strand. And using these together, we can make a single-strand cut here and a single-strand cut here, for instance. And this now, if you think back to what we were doing with genetic engineering in the human gene products, where we wanted to cut the DNA and leave the overhanging sticky edges, what you wind up here is you cut off the DNA here and you cut it here, and you wind up with now two pieces that have overhangs. And this might be of use in doing recombinant DNA technology. The other thing you can do is disable both of the nuclease domains in Cas9, resulting in something called a dead Cas9. You can take this dead Cas9 and you can add other domains onto it. What do they call here is heterologous effector domains. These might be domains that inhibit transcription, or these might be domains that accelerate transcription. or this just might be a fluorescent protein that allows you to localize something. And so the dead Cas9 that can't cut it all, because we're finding ways that we can attach other types of effector molecules to it, might allow us to better explore the genome, might allow us to affect not the genome per se, but transcription of particular genes. And that's where there's a lot of research activity today in further extending CRISPR-Cas9. Okay, so that is your overview of CRISPR-Cas9. What I want to do now is kind of give you a high level of some of the places where CRISPR-Cas9 is being used to treat infectious diseases and to treat cancer. Then I want to go back to muscular dystrophy and kind of take you through a chronologic process of where the gene editing clinical trials have gone and are envisioned to be going, to see if we can ultimately affect a cure for that disease. So CRISPR trials, and by the way, if you're really interested in these trials, at the bottom of the slides is the actual trial identifier for the trials we're going to be talking about. It was found that patients or the people that have mutations in the CCR5 gene appear to be resistant to HIV. If you have a heterozygote mutation, you appear to be somewhat resistant. If you have a homozygote mutation, you appear to be much more resistant. I don't know if you're completely resistant. Experiments revealed that the HIV virus not only has to bind to CD4 receptors on T helper cells, but also has to bind to the CCR5 protein receptor, which is the product of the CCR5 gene. And mutations, again, in that gene interfered with binding of the virus, thereby interfering with the ability of the HIV virus to infect these cells. And so there's a trial going on now where of course, they're taking donor hematopoietic stem cells. It wouldn't do very good to take autologous stem cells because they could already be infected with the virus. So they take donor hematopoietic stem cells. They're not taking CD4 cells, rather they're taking cells that are CD34 positive. This is a marker that's seen on hematopoietic stem cells and certain progenitor cells. And they're using CRISPR ex vivo to ablate the CCR5 gene with the idea that then they would infuse these stem cells back into patients. They would, of course, have a survival advantage. They would be resistant to HIV infection and possibly even affect a cure of HIV. And so this is an ongoing trial using CRISPR technology to treat HIV. You may recall from our cancer genetics discussion, we talked about PD-1 and the PD-1 ligand and the use of checkpoint inhibitor medications. When a T cell tries to kill a tumor cell, sometimes the tumor cell will manifest PD-L1, a ligand that basically serves as a checkpoint and basically tells the T cell, leave me alone, I'm a friend. And so the anti-PD-1 therapy like Keytruda has been developed in order to get around the tumor cells trying to inhibit that process. So there are several clinical trials now underway trying to take these T cells and actually knock out the PD-1 gene altogether. And so they harvest T cells from, you have a patient that has advanced cancer. You actually remove their T cells. You use CRISPR ex vivo in the lab to actually knock out the PD-1 gene. And then these T cells will be reinfused back into the patients. These are also ongoing trials using CRISPR for cancer. Every once in a while in the press, you might hear something about CAR T cells. CAR T stands for chimeric antigen receptor T cells. And basically what you're doing is you're taking a T cell and you're genetically engineering it by adding a new T cell receptor onto that cell so that that T cell will then attack the target of that new T cell receptor. And this picture, I took this picture out of a Science article. It doesn't completely align with this clinical trial. In the particular clinical trial, what they're doing is they're harvesting autologous T cells. So they're taking T cells out of the body. They're genetically engineering them using CRISPR. They're doing 3 types of things to these T cells. Number one is they're adding a T cell receptor that's specific to a protein that often occurs on cancer cells. Okay, this happens to be a protein that was discovered to occur commonly on cancer cells and appears to be one of the most immunogenic cancer antigens yet detected. So they introduce a gene into your T cells. That gene codes for T cell receptors so that then these T cells they're chimeric, if you will. They have chimeric antigen receptors because they have antigen receptors from two different species. So they're two different, not two different species, two different people. So they're chimeric. These T cell receptors are directed against this protein. The other thing that they're doing in this particular study is they're also knocking out PD-1, as in the prior slide, but they're also knocking out the endogenous T cell receptors because they find that's more effective. And so this CAR T cell therapy, which is also being explored for autoimmune diseases, basically you're taking T cells out or you're harvesting T cells from a healthy donor. You're either searching for tumor neoantigen candidates, or in this particular case, we know specifically what neoantigen we want to target. We're genetically modifying the T cells, creating genes, introducing genes that will produce new T cell specific receptors, then reinfusing those cells back in with the hope that there will be, an immune response, that these T cells will then kill any cells that are manifesting this particular receptor, whether that be cells that are producing, the cancer cells, or the lineage of cells stimulating the autoimmune disease, for instance. Okay, so that's some of the CRISPR trials out there. Now I want to return back to muscular dystrophy. And before we get to the CRISPR applications of muscular dystrophy, I'm going to kind of sort of do a little bit of a historical, it's funny to say historical. It's not like this stuff has been going on for more than a couple of years, but people's imaginations are flying, and the understanding of what all is feasible is rapidly maturing. And so I'm going to kind of present this as a little bit of a history, even though a lot of these trials are still ongoing, and the conclusions are yet to be determined. But think back a little bit to the genetic basis of muscular dystrophy. Think back to these large deletions and really what's causing the problem. The problem is you have these deletions, but the deletions are resulting in a stop codon, or they're resulting in a frameshift mutation. Now, we also remember, looking at the genetics of muscular dystrophy, that a lot of the protein, the critical functional pieces of the protein are at either end. And in the middle of that protein, we have these large repeat units. So the thinking has been, well, if there's a mutation in this particular exon, what if we just completely bypass that exon altogether? Just bypass that exon altogether, stitch together exon 3 and 5. We wind up with a protein, it's not a completely normal protein, but maybe it's partially functional. And so there's been experiments with what are called antisense oligonucleotides directed against exon 51 mutations. And when the pre-RNA is further processed to the mature messenger RNA, the antisense oligonucleotide induces exon skipping, and you wind up getting a messenger RNA where the mutated exon has been completely skipped. That then produces a partially functional dystrophin protein, and can restore, from Duchenne's muscular dystrophy, even if it makes you more like a Becker's muscular dystrophy, we can see a partial function restored. The other thing we see is that patients with muscular dystrophy are not necessarily dying from skeletal muscle pathology. They also can be dying from cardiac pathology, from congestive heart failure. So there is another study where allogenic stem cells are being injected into patients via coronary arteries just to see if they'll take up. And there's several of these cardiac stem cell studies, and really the focus there is to say, can we inject, these aren't genetically modified stem cells, we're just trying to see whether or not stem cells can be taken up, and what will happen to them. Will they actually grow, divide? Can they replace areas of damaged myocardium? Okay. There have been a couple of in vivo gene therapies for muscular dystrophy where the follistatin gene has been introduced or a microdystrophin gene has been introduced. The follistatin gene, the FST gene, produces follistatin, which inhibits the myostatin pathway, which inhibits muscle growth. So the notion is if you can increase the amount of follistatin, then you can further inhibit myostatin, and therefore the inhibition of muscle growth goes away, and you can stimulate, therefore, more muscle growth. And so the study assesses in vivo gene therapy feasibility. Can we actually, and what they're doing is they're injecting this gene into various muscles. These are genes that are carried in viruses, and they're just injecting them into various muscles, and they're trying to assess, is in vivo gene therapy even feasible, and can we find any evidence that gene therapy can improve function? And in this study, we can't inject a complete dystrophin gene. It's just too big. But we also know that the critical portions of the dystrophin protein are at the two ends. So what if we chop out a lot of the middle and we make a microdystrophin gene or a mini dystrophin gene? And so there's a trial, again, these aren't CRISPR-based. This is a trial where we're introducing microdystrophin. So there are mini dystrophin and microdystrophin studies that are also in vivo injections of a virus into muscle groups, and they're also assessing the feasibility of in vivo gene therapy and whether also trying to get confirmation in humans that a partial DMD gene is better than no DMD gene. Okay. We look at the use of CRISPR for DMD in some fairly landmark studies. There was a series of 3 studies that appeared in Science last year where they demonstrated that CRISPR could be used. If you remember, CRISPR is very good at cutting, but a little bit unpredictable at exactly what the nature of the mutations will be at the cut sites. But you can also use CRISPR, and what they did here was they used CRISPR in two places because really what they were trying to do was remove exon 23. These were mice that had a stop mutation at exon 23. So they wanted to see if they could use CRISPR and they applied the guide RNA that it was targeted to an intron segment between exons 22 and 23, and they supplied a separate guide RNA that was specific for the intron segment between exon 23 and 24. What they did was, you also have these size limitations with the adeno-associated virus, so they actually had to supply multiple viral vectors, right? They had to supply a viral vector that had the Cas9 gene with its promoter. They had to supply a separate viral vector that had a guide RNA for one of the intron segments, and then they also had to supply another adenoviral vector that had a guide RNA excuse me, for the other segment. So they had to supply 3 different viral vectors, which, is a little bit challenging because you need a cell to take up all 3 of those vectors. In this particular case, they also included a gene for a green fluorescent protein. They used that as a marker to see where the gene was actually taken up. And what you can see here, after the intramuscular injection at 3 weeks and then at 6 weeks, was how the level of green fluorescent protein increased. And down here in the staining for dystrophin, how the staining for dystrophin increased between 3 weeks and 6 weeks. And they did next generation sequencing, and they found evidence of, in fact, that exon 23 was skipped. Now, it's kind of a complex process that I tried to summarize here for what they did. They're not introducing, so the gene that's broken is the DMD gene, but they're not introducing a DMD gene. Rather, what they're doing is they're introducing CRISPR genes and CRISPR guide RNAs that are directed against the DMD gene. They're trying to alter the DMD gene. So they put the CRISPR genes into viruses. They introduced the viruses into muscle cells in vivo. The muscle cells then transcribe and translate the CRISPR components. It's the CRISPR components that then disrupt the DMD gene, and you wind up with a partially restored dystrophin protein. So this was really landmark evidence that CRISPR could be used. If you look at patients with Becker's muscular dystrophy, they have a much milder phenotype than those with Duchenne's muscular dystrophy. So while this is recognized as not being a cure, at least you can take someone with a more devastating phenotype, and there's hope that we could then convert that into a milder phenotype. And so one of the big researchers who's been doing a lot of the in vivo gene therapy in January was awarded a $2.2 million grant to explore gene therapy in Duchenne muscular dystrophy. And all we know at this point, at least all I could figure out at this point, is that he's planning to explore CRISPR-Cas9 technology as an approach, but there's not an actual clinical trial that's been announced or that's enrolling patients at this point. There's a group at UCLA that's thinking perhaps even more futuristically. Now, they're not anticipating this vision to come to clinical trial for maybe another 5 to 10 years, but in my mind, take everything we've just talked about over the last 2 hours, take everything and